Okay, so good morning, everyone. My name is Oshrat. I'm the head of the Translational Research Lab at the Asuta Medical Center in Tel Aviv. And today I will talk about the biology of Erdheim-Chester disease, or in short, ECD. So ECD belongs to a group of rare diseases called histiocytosis. And histiocytosis refers to a condition involving histiocyte, which is just a type of immune cell. And the word comes from Greek origin, in which histio means tissue and cyto means cell, and asis means too many or excessive growth. So basically, histiocytosis refers to an abnormal buildup of histiocyte, of these immune cells in tissues. And these cells, they normally help fight infections and clean up waste. But when they build up too much, they can damage healthy organs. So you can think of histiocytes just like janitors in school. So normally they clean up everything and keep everything running smoothly. But in histiocytosis, the janitors, they just don't stop working. So instead of removing the trash, they pile it up in the hallways. And over time, this clogs the school, making it hard to function. So the meaning is that histiocytosis occurs when histiocytes, these immune cells, they proliferate and accumulate where they shouldn't. And this leads to inflammation and tissue damage. So the accumulated histiocytes in tissue, they release what we call cytokines and chemokines, which are just small proteins in the body that the body uses to send messages to recruit even more immune cells into the tissue. And since these histiocytes, they release too many of these messages, too many of these cytokines, it leads to chronic inflammation and damage the healthy tissues. So before targeted treatments were discovered, the main way to reduce inflammation was using drugs that blocked cytokines. So chronic inflammation due to excessive cytokine release, it can cause pain. And this is usually the first symptom that leads patients to see their doctor. So in ECD, the histiocytes, they accumulate in various organs, including the bones, the heart, lungs, kidneys, and brain. And this infiltration disrupts the normal organ function and causes this chronic inflammation and pain that I just mentioned. So in order to understand disease biology, we need research. And basically, we have three main types of research. So the first one is what we call basic research. And in basic research, we are investigating the biological mechanism of the disease. We're doing studies in animal models and in cells and tissues. These are the laboratory models we have to study the disease. We're doing genetic testing and molecular analysis. The second type of research is what we call clinical trials. So in clinical trials, we are evaluating new drugs on patients, and these studies are usually based on the laboratory findings, okay? And I will give you an example in a second. The goal is to evaluate potential side effects of new drugs as well as how effective they are. And the third type of research is what we call data research. And in this type of research, we are collecting data for clinical insights. So we are using medical records and registries, and we are analyzing the collected data in order to improve outcomes. So today I will focus on these basic research studies and how discoveries that made in the lab led to successful clinical trials and improved patient outcomes. So when you think of a research lab, so it's just like a bridge, connecting the basic science to the clinical practice. So the meaning is that discoveries start in the laboratory settings, they are developed into potential treatments and reach ECD patients in clinical practice. So you can think that just like as a building is made of bricks, so our human body is made of cells. And inside each of our cells, there is the genetic material, what we call the DNA. So the DNA, it contains the complete instruction needed to create a healthy human. So DNA is a very long sequence, consists of 4 letters, A, T, C, and G. And it's just like a language, okay? And in this language, every 3 letters, they form a word. And a group of words, they form a sentence. We call it a gene, okay? And the sentence, the gene, gives instruction to the cell to make proteins. And proteins, they instruct the cell what to do and how to work properly. So today in the lab, we can read our entire DNA from beginning to end, and changes in one of the letters, changes in this language, in this sequence, in a significant region of the DNA can lead to diseases. So these changes are what we call in the scientific language mutations. So what is a mutation? It's just a small change in a significant region of our genetic code, of our DNA, and it's like a typo, replacing a letter in a word, changing the meaning entirely. So, for example, I can have a book and a cook, and I only replaced B with a C. That's just one letter, but it turns into a completely different word with a different meaning. So a change in the DNA sequence can impact a biological mechanism, because now the sequence is no longer understood correctly. So how does a mutation affect a biological mechanism? Well, inside our cells, there is a normal transfer of information. We have our DNA, our genetic code, and just a small unit of information from our DNA is converted into a molecule called RNA. These are the genes, okay? So then the RNA is used to make protein, and as I mentioned before, proteins are what actually do the work in our cells. They control how our cells grow, how they divide, how they function. And when a mutation occurs in the DNA, it disrupts this flow, and this can impact a biological pathway, such as those controlling cell divisions. So how mutation changed ECD treatment? So for many years, ECD was truly a medical mystery. Physicians, they saw patients with abnormal immune cell growth, but they didn't fully understand why it happened or how to treat it effectively. So that changed dramatically when researchers discovered that many patients with ECD have a mutation in a gene called BRAF. And this mutation leads to the activation, as Dr. Goyal said, to activation of a biological pathway that we call the MAP kinase pathway. And this leads to abnormal cell growth. So in fact, over the years, researchers have found additional mutations in genes that activate this MAP kinase pathway, genes like MAP2K1 and RAS and others. And when we have a mutation in these genes, it leads to abnormal cell growth. And these cells release the same cytokine, I was talking about them in the beginning, that trigger the chronic inflammation. And this combination is what truly drives disease progression. So the conclusion from the basic science research was that mutations in the MAP kinase pathway, they drive disease progression. So what can we do in the clinical practice? So based on these laboratory findings, clinical trials were initiated in patients, first with BRAF and later with MEK inhibitors. And this treatment, the BRAF and MEK inhibitors, they aim to block this overactivation of the MAP kinase pathway, helping to reduce inflammation and slow the disease progression. And these studies led to FDA approval of BRAF and MEK inhibitor for the treatment of ECD, which I'm sure that some of you are being treated with. So today we have new clinical trials testing new biological drugs, based on different mutations that were discovered. So these trials are ongoing in multiple sites all over the world, and we are still waiting for the reports. And what I want to show you also is that in recent years, beyond this DNA mutation I just discussed, some progress has been made in a field called epigenetics. So epigenetics controls whether genes are turned on or off, but without changing the DNA sequence. So we can have a gene that will be expressed or shut down, but there will be no change in the sequence of the DNA. The sequence is still read correctly. So while MAP kinase mutations play a key role in driving the disease, we see that changes in gene expression without altering the DNA sequence may also contribute to ECD progression. And this is just one example. This is what we call in a scientific language DNA methylation. So DNA methylation is the addition of a chemical group named methyl, and this chemical group is on top of the DNA. We don't have any change in the DNA sequence. We just put this methyl group on top of the DNA, and this is DNA methylation. And this DNA methylation can determine either a certain gene will be expressed or shut down. So when we have this addition of this methyl group, this DNA methylation, on top of the DNA, this gene will not be expressed. It's just like a stop sign. And we found that a specific gene named MIR-LET7B, this is the name of the gene, is methylated in patients with histiocytosis. And this is ECD, LCH, and Rosai-Dorfman disease patients. So this gene is being methylated, and since this gene is methylated, it has this stop sign. It cannot be expressed, and because of that, it does not generate its mature form, which the mature form is just a small molecule we call microRNA, so it's called LET7B microRNA. So the gene is not being expressed, the mature LET7 cannot be formed because he has this stop sign, and what we found is that indeed patients with histiocytic neoplasm, bless you, have low levels of LET7B microRNA compared with healthy controls. So why is it important? Because in normal cells, this LET7B microRNA is expressed and it suppresses key genes in the MAP kinase pathway, the same pathway we just discussed that is overexpressed due to mutations. But here we don't have a mutation. In ECD, as I showed you, this LET7B is downregulated, so its expression is being reduced. And as a result, it can no longer suppress the MAP kinase pathway. So this leads to MAP kinase overactivation, even in the absence of DNA mutations. So why it is important? Because even if a patient doesn't have a mutation in their DNA, changes in gene activities, these epigenetic changes, can still turn the MAP kinase pathway on. And this means that some patients without mutations in their DNA may still benefit from the drugs that block this pathway. So what we also identified is that this same LET7 microRNA, it keeps a gene named BCL-XL under control. And BCL-XL, this gene, it helps cells stay alive and grow, okay? So what happened when this LET7B is missing, because I showed you that it is downregulated in ECD patients. So when this LET7B is missing, it cannot control this gene expression, this BCL-XL. And as a result, BCL-XL gene becomes too active. And this may cause the cells to survive and make the disease even worse. So we did confirm this in tissue biopsies from patients with ECD. These are just 3 examples of tissue biopsy, and we see here that BCL-XL is highly expressed in this tissue. And how do we see it? You see these brown spots. So every brown spot is a protein expression of this BCL-XL, and you can see it's all over the tissue. And what we also identified is another gene that works together with this BCL-XL. You don't see it here in the picture, but this protein is called BCL2, and it is also overexpressed in some of the patients. And why do we need it? Because today there are BCL2 and BCL-XL inhibitors already in use in hematological disorders or in clinical trials. And this supports the need for ongoing research into this treatment option, which may lead to new therapies in the future. So to summarize, we discussed that ECD arises from abnormal growth of immune cells called histiocytes, leading to inflammation and tissue damage. I showed you how genetic mutations, mainly in the MAP kinase pathway, drive abnormal cell growth, overproduction of cytokines, and inflammation. And understanding this mutation led to targeted treatments blocking this pathway. And other treatments are currently under clinical investigation. And I can summarize and say that ECD is characterized by a complex network of genetics and epigenetics mechanism. Some are already targetable, and some will be in the future. So by this, I want to say thank you, and I'll be happy to take any questions if you have. Yeah. To go back to the clinical trial slide. No problem, just a minute. This is the slides. Okay. I'll put back on the slides. Here, it's on the desktop, I said. Can I open it? Yeah, it's okay.
It's not projecting.
Mm-hmm. Oh, this one. Oh, but they're not showing. Okay, okay, now it is. Oh, welcome. Oh. Let's do it like this. Yeah. Okay.
Any other questions?
Yes.
Is there any research looking for, say, the reason why you have mutation? Anything, any suspect of what's happening maybe in the habits of the people that cause this mutation?
You mean like environmental factors and stuff like this?
that.
So basically, I think not enough patients to do these kinds of studies for now, but there is a big, what Gaurav just mentioned, that the Italian and the French one are summarizing all the cases, and they will look into it once they will collect enough data. Yeah.
Thank you.
No?